Okay, hello everybody. I'd just like to quickly talk through some of the key points from the lecture I gave on protein phosphorylation. So, protein phosphorylation is a common way the cell uses to modify the activity of proteins. So rather than having to make new proteins, the cell can modify the behavior of an existing protein. So in this little schematic here, we have a newly synthesized protein, or a protein that exists in the cell, and through the action of a kinase enzyme, this protein can be phosphorylated. So here we have a protein with a phosphate group attached to it, and conversely, a phosphorylated protein can be dephosphorylated by an enzyme activity called a phosphatase. Okay, so protein modification by phosphorylation is a reversible reaction, so the cell can use it to switch on and switch off the activity of pre-existing proteins. There's a simple example given in the textbook of how two different enzyme activities, such as glycogen synthase and glycogen phosphorylase, can be the activity of these enzymes can be controlled by phosphorylation. Now, in the case of glycogen synthase, shown here, this enzyme is inhibited by phosphorylation, and in the case here of glycogen phosphorylase, its activity is increased by phosphorylation. So phosphorylation activates the phosphorylase. So by having so effectively the cell is coordinating the metabolism of glycogen. So effectively the cell is controlling the uptake of glucose or the release of glucose from glycogen stores through the phosphorylation of enzymes involved. Now, for a protein to be phosphorylated, something has to donate a phosphate group. So here we have the ATP, a common cellular nucleotide, and it's the gamma phosphate group, it's the alpha beta, it's the gamma phosphate group here that is donated to the kinase enzyme, and that phosphate group is transferred to the protein. So there are two main groups of kinases in the cell. There are serine threonine kinases and there are tyrosine kinases. Now, the serine threonine kinases are able to add a phosphate group to the alcohol group on serine residues, and similarly the tyrosine kinase residues can add phosphate groups onto the OH group on the aromatic ring of the tyrosine residue. So proteins can be phosphorylated on many sites because serine, threonine and tyrosine residues are clearly very common in proteins. Some examples of different kinase activities in the cell could be things like protein kinase A, phosphorylase kinase, and some calcium-dependent kinases. Now, in this example here, each of these three different kinases can all target the same protein, which is glycogen synthase. So given that kinases are important activities within cells, then it's good to have a look at one of these kinases in a bit more detail. So protein kinase A is a good textbook example. So protein kinase A is a protein that's made up of 4 subunits, 2 regulatory units and 2 catalytic units. So here's a schematic here of protein kinase A with the 2 catalytic units in blue and the 2 regulatory units shown here in grey. And upon activation of protein kinase A, then the catalytic subunits are able to break away from the regulatory subunits. So protein kinase A is actually regulated by cyclic AMP. So 4 molecules of cyclic AMP can bind to the regulatory subunits to release the catalytic subunits. And then these catalytic subunits can go away and phosphorylate proteins within the cell. Another example of a regulatory kinase in the cell is protein kinase C. Now, protein kinase C is not regulated by cyclic AMP. Protein kinase C is regulated by the combined action of diacylglycerol and calcium ions. Another example of a protein kinase in the cell is a G protein coupled receptor kinase. These kinases can phosphorylate the G protein coupled receptors. So if you remember, we looked at the receptors in the last lecture. The receptors have a carboxy terminus on the inside of the cell. These kinases can phosphorylate residues on the carboxy terminus of the G protein coupled receptor. And once you phosphorylate the receptor, it's no longer able to function as a receptor molecule, and it modulates the activity of the receptor. So if you recall, earlier I said there were 2 broad classes of kinases. There were the serine threonine kinases, and there's also the tyrosine specific protein kinases. Now we're going to have a quick look at these tyrosine specific protein kinases. Now, there are 2 broad classes of tyrosine kinases. There are those which are part of a receptor and which are membrane bound, and there are those which are soluble. Okay, this is just showing the motif on a target protein that contains a tyrosine residue that can be phosphorylated by these tyrosine kinases. So receptors that contain tyrosine kinase activity, there are 2 types. There are receptors that have an intrinsic tyrosine kinase activity, meaning that the tyrosine kinase domain is a part of the same protein that is actually the receptor. Okay? And these are referred to as receptor tyrosine kinases. So this is like a little diagram here which shows the receptor tyrosine kinase, where the tyrosine kinase domain is a part of the actual receptor. There's also receptor tyrosine kinases where the kinase is an associated protein. So the receptor itself doesn't contain a tyrosine kinase domain, but there's an associated tyrosine kinase domain with that receptor. And we'll look at some examples of these. We've looked at the activity of these receptor tyrosine kinases before, and if you recall, it's the ligand binding to the receptor that stimulates the catalytic activity of the receptor. So effectively, ligand binds, it activates the tyrosine kinase domains, and then that will effectively lead to autophosphorylation of the receptor, and this creates binding sites for effector proteins. It's generally assumed that the autophosphorylation of the receptor that occurs upon ligand binding occurs by a trans mechanism, meaning that one of the chains of the receptor performs a kinase activity on the neighboring chain. So it's a transformation. Now, once you've phosphorylated the receptor, once it becomes phosphorylated, these phosphorylated tyrosines then provide binding sites for other signaling molecules, and effectively these phosphorylated tyrosines provide docking sites for other proteins to bind. And here's a little schematic here from the textbook showing the epidermal growth factor, the ligand binding to the receptor to form a receptor, a functional receptor, in which the intrinsic tyrosine kinase activity can phosphorylate the other chain, whereas this tyrosine kinase can phosphorylate this chain, leading to phosphorylated tyrosine residues on the receptor, allowing other proteins to bind to the phosphorylated tyrosine. And as we'll discuss in a minute, it's an SH2 domain of an adaptor protein that can bind to a phosphorylated tyrosine, or it's the SH2 domain of another protein which may have an enzyme activity that can bind to a phosphorylated tyrosine. And in the example we look at in the textbook, the adaptor protein GRB2 can also bind to the SOS protein through another binding domain to propagate and create a signal transduction pathway. Typically, the SOS enzyme is able to activate a RAS molecule, which can then activate a kinase pathway referred to as the MAP kinase cascade, leading to gene activation. Now, there are other proteins that contain SH2 domains as well. An example would be phospholipase C gamma, and phospholipase C gamma can bind directly to a phosphorylated receptor, and then that can activate the lipase to cleave membrane lipids which leads to another signaling pathway, which we'll discuss in another lecture. So, as we mentioned earlier, the GRB2 protein is an important adaptor protein. So GRB2 can bind to a phosphorylated tyrosine on a receptor molecule, and it forms part of the molecular Lego, if you like, to a molecular building block, so that other proteins that can't bind to the receptor can now bind to the GRB2. Examples of binding partners of the GRB2 adaptor protein include the epidermal growth factor receptor, the platelet-derived growth factor receptor, and a bunch of other receptors. But GRB2 can also bind to the SOS protein, and we've seen that in the diagram where GRB2 acts to bring the SOS protein to the receptor. So the GRB2 acts as an adapter, since SOS can't bind to the receptor in its own right. So looking at this idea of adapter molecules, just very briefly, here's a few adapter molecules. So here's the GRB2 molecule, and we also discussed the IRS1 molecule, the insulin receptor substrate molecule, when we look at insulin. Both of these adapter molecules contain binding sites for other proteins, but neither of these contain functional enzyme activities. So they're adapters because they can bind and bring together proteins, but they don't act as enzymes in their own right. So So the SH2 domain is a common domain that occurs in many proteins. And proteins that contain SH2 domains may be able to bind to receptors, and these proteins also become targets of phosphorylation. And therefore, when the SH2 containing protein is phosphorylated, it then presents binding sites for other proteins. So in this way, you can link together various components of pathways through these adapters. Okay, so we've been looking at tyrosine kinase receptors. There's also another class of tyrosine kinases that are soluble. So these are the non-receptor tyrosine kinases. So these are located in the cytoplasm, and they can be associated with receptors, but they are not receptors themselves. There are two examples of non-receptor soluble tyrosine kinases that we look at. One of them is the Src kinase, and the other one we'll look at a bit later are the JAKs proteins. So the SRC protein can interact with a bunch of different receptors, and it was actually the first receptor tyrosine kinase discovered. The SRC kinase is shown here. It's an interesting molecule in terms of its regulation because it can auto-regulate itself. So if you look here, SRC has an SH2 binding domain, and SRC has a tyrosine residue that can be phosphorylated, and it has a tyrosine kinase domain. So effectively, the SRC kinase can phosphorylate the tyrosine, and then it can bind to the SH2. So this is shown here, where the tyrosine of the SRC is phosphorylated by a tyrosine kinase, and then the phosphorylated tyrosine is bound by the SH2 domain. So effectively, the SRC kinase protein can auto-regulate itself, and in this form, it's inactive. There are other effector proteins for SRC kinase, and these effector proteins typically can have an SH2 binding domain that binds with a greater affinity to the phosphorylated tyrosine than this SH2. So effectively, the stronger binding of the SH2 domain on another protein can displace and open up the SRC so that the kinase activity is active. So that's one of the ways. There are other methods as well, but that's one of the ways that SRC can become active. Because in this inactive form here, the kinase is hidden from its substrates. Whereas in this open form, the kinase activity is able to phosphorylate other proteins. Another class of soluble tyrosine kinases are the JAKs proteins, and we look at the JAKISTAT pathway in the textbook. So here's the JAKISTAT pathway. Here's a cytokine ligand that binds and causes dimerization of the receptor. Again, we have the JAKs, which are the soluble tyrosine kinases, coming together at the receptor or are associated with the receptor, and then the JAKs proteins can trans-phosphorylate the receptor in the same way that the intrinsic kinase activity of the receptor tyrosine kinases could trans-phosphorylate the receptor. So this is very much like a tyrosine kinase receptor, except that it's an associated kinase activity to the receptor. Once the receptor is phosphorylated by the JAKs proteins, these phosphorylated tyrosine residues then form binding sites for these STATs proteins, and these STATs proteins become phosphorylated by the receptor, and then they can dimerize, and then they can be transported to the nucleus to activate gene transcription. And one of the products of the gene transcription, apart from effector proteins that carry out another process involved in growth or differentiation and all that kind of thing, one of the proteins that can be transcribed are the SOX proteins, which stands for suppressor of cytokinin signaling. And these SOX proteins can effectively suppress signaling from the activated receptor. So these SOX proteins also contain SH2 binding domains. So lastly, I'd just like to mention the MAP kinase pathway. So at some point we'll be talking about activation of RAS, and one of the main effector proteins of active RAS is the RAF protein. So RAF is the first stage of a MAP kinase pathway. So the RAF is referred to as a kinase. So it's the upper level of this MAP kinase pathway. RAF is able to phosphorylate the MAPK, and then MAPK is able to phosphorylate MAP kinase. So we have a cascade of 3 kinases. The top level kinase is a serine threonine kinase. The second kinase in the pathway is a tyrosine threonine kinase, and the MAP kinase itself is a serine threonine kinase. So as we've stated earlier in this lecture, we have a transduction pathway whereby the receptor activates the RAS, and then the RAS can then activate the MAP kinase pathway, which is what we're looking at now. And the MAP kinase pathway is highly conserved across eukaryotes, from yeast through to mammals, and the typical pathway you'll come across in mammals in the textbook is the RAF MEK1 ERK1 pathway. So a typical example of a MAP kinase pathway is RAF MEK ERK, and this is just a little schematic here showing a couple of these MAP kinase pathways from different species. Here's the yeast example here, where we have the 3 kinases held together by a scaffold protein, and here's an example where the upper kinase actually forms a scaffold for the 2 lower kinases as well. And just very quickly, I'd just like to point out that there's a whole bunch of kinases that we've been looking at, these tyrosine kinases and other kinases. It's also important to be able to turn off signaling through kinase activity, and this is typically carried out by phosphatases. And there are so the receptor tyrosine kinases their activity can be controlled by phosphatases that remove the phosphate group from the tyrosines. And don't forget, we've also discussed other off switches for signaling, such as endocytosis of membrane proteins to attenuate the signals. Okay, well, I hope that lecture was of some use to you. So good luck. Thanks.